One piece in the story of the genetics of idiopathic Parkinson's disease is based on a gene called LRRK2. The genetic-based form of the Parkinson's disease is typically starting 10 to 15 years earlier compared to the normal manifestation age, which is in the range of about 65 to 70. The genetic-based are in the range of 50 to 55, and therefore there's always an interest to understand why these early manifestation patients have any specific environment, makeup in the genetics that can explain that they are starting earlier with the manifestation. ROPAD is an international global study where we are enrolling 10,000 patients suffering from so-called idiopathic Parkinson's disease. The study is clearly focused on the patients. It means we're offering the patients, again 10,000 patients, a huge number, not only a standardized diagnostic platform, where at least in some countries it might be difficult for the patients to get access to this genetic platform. We are also allowing the patients to be enrolled in a prospective longitudinal monitoring way. The 10,000 patient cohort is definitely the world's largest cohort that is planned to be analyzed from the genetics, but also from the very sophisticated, deep clinical investigation perspective. More important, that the ROPAD has a very strong scientific background in the sense of what are the very clear manifestations of the patients, what are the first symptoms, how the patients start to develop Parkinson's disease. Can we predict the progress using the proteomic metabolomic platform to characterize specific biomarkers that hopefully will give us the chance to improve the overall prognostic prediction for those patients on the one side, and on the other side, ideally use any of these biomarkers also to demonstrate efficacy of treatment. Whenever you identify the target, you might have an idea how to treat these patients in the future in a better way. The better we characterize the patients, the easier it is for us as a company, but also as scientists, to develop specific biomarkers that can be used for the prediction of the progress or as a parameter demonstrating the improvement once the patients are being set on treatment. There's a further element because more and more pharmaceutical companies are getting active in the development of inhibitor of the hyperactivated LRRK2 kinase. We are also contributing that the development of these drugs is speeding up because first you have to identify the patients, and once you have identified the patients, you can check whether the patients might be eligible for such clinical programs.